I'm going to show you how to run the Chromatin Structure Analysis Workflow here in the FormBio platform. I'll assume that you already have logged in and are in the project that you want to run your analysis in, and that your data is also already uploaded to the same project. So I'm going to go to the Launch Workflow and select Chromatin Structure Analysis here on the top right. In this overview page, you have information of which data types you can analyze using this workflow and which are the steps comprising the analysis for each of the data types, as they are different. You also have information of which inputs are required for setting up the workflow and which specific data formats we might need. On the Outputs tab here, we have information of all of the different types of files that are generated by the workflow and which data they actually contain. And we have citations for the relevant papers and tools that we are using. We're going to start setting up our analysis by clicking the Run Workflow here in the top right corner of the screen. In case we want to redo a previous analysis or an analysis using a different version of the workflow, we can also select that from this drop-down menu here. Once we are in the Workflow Launcher page, we can start by selecting which data type we want to analyze. In this case here, I'll be using ataxic data, so I'll select that here. I'll be using data that was sequenced using the Illumina technology, so I'll keep that. And I'll be doing a full analysis, which includes QC of the data, the read mapping, and the peak calling. I could also do the alignment only, or if I already had BAM files or CRAM files available, just use those as input and do peak calling only. I want to map my data to the mouse genome, so we will select that here from this drop-down menu, and just by scrolling, you can have an idea of the genomes that we have available in the platform at the moment. Then I'm going to select the directory where I have my FASTQ files. So I click the Select Directory button and I have them here in this ataxic. Just click Submit. And the next thing that I need to do is to create or load a sample attribute file. I already created one previously, so I'll just click Select Existing. That one is in the same directory where I had data, is this design.txt file, so I'll select that and submit. It's going to open up a table that shows what is inside of my file. The run IDs, they are detected or identified from the names of my FASTQ files. I have my sample IDs, as this is just two replicates from the same tissue. And I don't have controls IDs, so I just copy the sample ID to the control ID. If I had controls, I would add the sample ID of my controls to the matching non-control samples. If I was creating the file from scratch, I would add the name here in the file name and click the floppy disk to save this design file. And this file will be saved in the same directory where we had our input data. Clicking Next Step, we have some more configurations to do. I'll click Trim if I want the reads to be trimmed, and remove adapters, remove the poor quality ends and very short reads. I'll be using Trim Galore to do those filtering steps. I could select also Fast View and Cut Adapt. I want my alignment output format to be BAM, so I'll keep that. We also have the option of using CRAM, and those are usually required if you have genomes with very long chromosomes, for example, plant genomes or marsupial genomes. I will select Parabricks BWMM for doing the read mapping, but we have other options, Sentient BWMM or the open source version. Minimap, we have Sentient and Parabricks are usually accelerated, faster versions of the open source tools. So I would advise to use those if you have the option. I want my duplicates to be marked, so I'll keep that P card here. If I didn't want it, I just could skip this step as well. The last choices I can make are the minimum mapping quality for keeping my reads, and also the maximum peak length for max tree. I will keep this default as well. Then I go to the review page where I can check that all of my inputs and all my selections are as I want them to be. And I'll end up by just giving a name to my run and click the Run Workflow button. Now that our workflow is running, we just have to wait until the analysis is complete. I already completed an analysis previously, so we won't have to wait here. So I'll just click the line here, and this is going to take me to the results page, where I'll have access to the multi-QC report right away. I'll click this link so we can see it in full screen. We have information from QualiMap, vCard, some tools, CutAdapt, and FastQC. This might change according to the selections that you made during the setting up of the workflow. You can take a look at all of those and make sure that everything looks okay with our data and the results. We have other HTML pages here for the FastQC results, separately for our samples and QualiMap reports as well. We also have many other files here with plots or statistics. Because these are so many files and we need to scroll quite a lot, I usually like to go just to the All Files tab, where these files are within folders, so it's easier to get what we want. And then click the Output folder, that's where we have all of our results. And again, you can see the HTML for the multi-QC report. We have some QC metrics for our data, for example, read counts. We have profiles and fingerprint plots here. We have our alignment results here, band files, and also some QC metrics associated with them. The browser folder, I think, is one of the most important ones for this type of analysis. Is where we have all of our bed files and the gap-pick files that actually have our open chromatin regions. These files, any of them, you can click them. Some of them will be able to visualize them if they are small enough, and if we have like a PDF viewer in here, we can download them directly to our laptops. And we can also copy them within our project to a folder that we want for our choosing, so we can keep everything organized. And if they are compatible with visualization with IGView, there's an app button that will appear here, and we can click it and visualize it in the browser too. Other data we have here includes the status of each process that was run during the workflow. In our case, all of the status are completed. We didn't have any issues. If something didn't go as planned, we would have a completed with warnings or a failed status here. And we could go through each of the processes output files by clicking this plus button here to be able to debug. We also have any of the Nextflow logs here available if we want to debug further. We also have access again to all of the inputs and even some of the default inputs that are used in the workflow. So we have this information, for example, if we want to write a publication in the methods section. And these are the main results, the main files that we obtained by running the Chromatin Structure Analysis Workflow.